4:35. We probably should get started here. So it's a real pleasure. I'm Simone Schnall, and it's my real pleasure to welcome Hilke Plassmann today. She's a professor, a chaired professor at the INSEAD Business School. And she's really at the forefront of the field of neuroeconomics and neuromarketing. So first of all, her precise job title is the Octave Pharma Chaired Professor of Precision Neuroscience. And she also has an appointment at the Paris Brain Institute. So she does really innovative work that combines psychology, marketing with economics, neuroeconomics, and so on. So covers a lot of ground, very exciting work. And I was just mentioning to Hilke, I came across her work initially when I was talking to Wolfram Schultz, who's here at Cambridge and who's done a lot on the reward system and how single neurons play a role in that. And I asked Wolfram, so, what should I read about the reward system? And he said, well, look at that paper by Hilke Plassmann. And that was a very cool paper, 2007 or 8, showing that when you tell people that wine is really expensive, first of all, they like it more, but then also the brain's reward structures reflect that it's more valuable or more precious to them, more rewarding, basically. So anyway, so Hilke has done a lot of really cool work, and today she'll be talking about individual differences in decision-making and how they come into interactions between the brain and the gut, and the roles of that, or the impact of that on behavior as well. So thank you very much, Hilke. Please take it away.
Thank you, Simone, for this very kind and nice introduction. Wolfram is, we think of him as the grandfather of neuroeconomics. And when I did my postdoc at Caltech, and this is when I met him, and this is also where he got his inspiration for his really seminal work in neuroeconomics, chatting with economists, right? So, and I remember quite well our interactions. So say hi to him when you see him. And yeah, thank you so much. I'm so honored to be here. It's a big pleasure. And so I decided to talk about some work that is actually not that linked to marketing, but much more fundamental in a sense that I've always been interested in. So I'm fascinated by human behavior, and I do think that it's very valuable to look at individual differences to understand really behavior, but that sometimes we have all these scales about personality traits, which is important, but I also think that there's some other thing happening, and we see it actually quite nicely with COVID right now. So some people, they're vaccinated and whatnot, and they get COVID, and other people, they never got it. And so there's something, I think, in the composition of our biology that might have more explanatory power than we think, right? And I'm specifically interested in consumer decision making, specifically if they make disadvantageous decisions. A lot of my work looks at food decisions. This would include the wine, but also if people make trade-offs between healthy and tasty items, what can actually explain why they choose something healthy or not, or also why they have impatience for rewards. So why are they not wanting to wait for larger monetary reward or not? But to motivate this, I was telling you in the coffee part that you will need my kids. So this is my oldest son. He's actually now, I should correct this, he's now 6 years. His name is Mats. And so I did the marshmallow test with him. Although they're German, they don't like marshmallows. They really like Kinder Schokobons. So I said, look, so here is a bowl of Schokobons. You can wait. If you wait, you can eat them all, or you can have one and eat it immediately. And he's quite relaxed looking at them and is not actually eating them immediately. Now, this is my youngest. His name is Janis, and so he already has a finger in his mouth, looks at them, and then I can't, it was hard to actually take those pictures because immediately he's eating them, right? So, and you see, so here, I mean, obviously there are lots of reasons, also because they have different ages, right? So, and the brain and specifically the console structures might be developed differently at their age, right? So, but we see here that although their genetic makeup is, very similar, they have very different abilities to wait for a larger later reward and so make a normatively better decision, right? And this is not unique, right? So this was when you think of the advent calendar, which I think is actually made as the test of delay of gratification. So Mats did wait, and every day one door. Janus ate all of the doors in one day. So because he could not wait. So. And so this is not a phenomenon that is unique to my children, right? So it actually is something that is quite well known, specifically in the economics literature. When we look at, we call this their delay discounting. So this means how much people discount the rewards, that the time that they have to wait for the reward to be available, right? So, and it's normal that I mean, so it makes sense because of inflation and uncertainty, right? That we do prefer rewards sooner rather than later. But what we see here, and this is what I'm interested in, is that for some people this is very steep, like you see for this person, and for this person it's very flat, right? So so these are only 3 individuals taken from a seminal neuroeconomics paper on this topic from Joe Cable and Paul Glimcher. What is interesting now is so that these individual differences do exist, and this now or later trade-offs, we actually face them in a lot of different types of decisions. So again, the behavioral economics version to look at this, and then what they label self-control is this idea of looking at monetary rewards sooner or later in time. But also you can think of, for example, food choices, right? So you can think of that there's a short-term reward of eating the chocobons now, or the long-term reward, which is that if I avoid repeatedly eating a lot of chocobons, I might be of better health, right, and might live a longer and better life. Same is also maybe true for even for social decisions, right? So there's always this idea of temptations, and should I keep money for myself, or should I, for example, donate it to other people? And this goes a bit to the idea of that this to people, or to this to self theory that I'm sure you're familiar with as social psychologist. And the same is so also true for delay discounting in these food choices, that I'm thinking of a person now versus another Hilke later in time. And these are the types of conflicts I'm interested in, and I'm interested in understanding how how using the brain, or how using other biological markers can help us to better understand my ability to make healthier or more patient choices. And so this is kind of a bit what summarizes this line of my research, right? So, and when you think of this, so I will start my talk talking about a lot of, a lot of people have done this work where we're looking at, okay, what are actually the brain systems encoding this, right? My work, as compared to other work in this area, has been characterized to look at some very specific differences in, for example, also the neuroanatomy of people. And this is kind of what I will want to start with. Is the first study where we look at, so I'm going to switch a bit back and forth between monetary rewards and also these types of trade-offs, right? So the Brussels sprouts where we'll see yummy chocolates. And so this first study that I would like to present is really asking a very fundamental question. Are there any differences in our neuroanatomy, so in our brain structure really, that could predict my ability to say more often yes to the Brussels sprouts as compared to the chocolates, controlling for whether I actually like the chocolate more than the Brussels sprouts? And this is joint work with Todd Hare, Henry Hutchison, Anita Tusche, and was spearheaded by my former postdoc, Liane Schmidt. And so these are the types of tasks. I'm going to explain it a bit more here because I will go back to this at a later stage, right? So what we do is we ask people to make decisions as they would usually do. And then we tell them, so you see in food item, you have to tell us how much you want to eat at the end of the experiment. At the end, we implement actually one of the trials. So if they say now they really want to eat the yogurt, and then the yogurt trial is drawn, then they really have to eat the yogurt, right? And so this is in some of the baseline condition. Then we have a condition where we ask them, now I want you to really think about the tastiness of the item. And then there's a third condition where we want them to consider the healthiness of the item. And then for this paper, what we were interested in is actually this part here, where we're looking at the healthiness of the item and whether we can predict in this health condition whether they're able to regulate themselves. And just to give you the intuition for what we have done here, so what you can do, given now the behavior, is you can think about what predicts best their answer. So this answer, whether they want to eat it, yes or no. So it's a strong yes, yes, no, strong no. So it's a 4-point kind of preference measure. And what we're seeing here is that, which makes sense, right? So is that taste rating has a big influence on this. So it's a very good predictor. So this is, sorry, I didn't explain. So we also asked about the subjective taste and healthiness of the item, sometimes before, sometimes after the main decision-making task, right? So we see here that the perceived tastiness predicts this, what they want to eat. We also see what is nice is that here's a dummy regressor for the condition. So we see that health condition, when I ask you to focus on health, they generally want those items less. In this specific experiment, they were usually quite unhealthy, so this is a good thing. Taste focus had a positive regression. What we're now really interested in, though, is our measure of self-control that we want to predict based on the neuroanatomy, is the idea that now, so in the health focus condition, in order to make a healthy choice, I need to either focus more on health or I need to focus less on taste, right? So this is what you see in these interaction terms that are in red here. And what you see also, they're both also significant. So people seem to apply these two strategies, right? So to come to make a healthier choice. And this together, we actually used as a behavioral measure of regulatory success, where we put these two together. So it's not one versus the other. We're looking at where it doesn't matter to us which one. We're interested in that they're able actually to make to focus on the healthiness. And so now what we're doing is, so we're looking, so this was in the first data set, which were 91 participants, where we're looking at, can we actually through a leave one in, leave one out prediction, can we actually predict this regulatory success score. And so in other words, we did a cross-validation. And so what we did here is also, we didn't look at the whole brain. We had priors, right? So based on actually all of our prior work using Fmri from the author team, right? So that there should be 2 brain systems involved, the valuation system that Simone talked about earlier. And specifically for the food items, I see very often this chunk here, the ventral medial prefrontal cortex. And then also what we often refer to as the control system. Obviously, you know, I'm doing a bit labeling here and reverse inference. So this would be the dorsolateral prefrontal cortex, what you see here. And so we looked at the gray matter density in those areas and looked at whether this would predict their regulatory success, again, using this cross-validation procedure. And yes, so what I'm showing you here is the predicted regulatory success in relation to the observed regulatory success. And we do find a significant correlation between the two. To push this a bit more, we actually then said, okay, now let's take this model so we can, you know, come up with a general linear model based on this first data set. Can we predict in a different data set, in a slightly different task? Now knowing about the importance of these two structures, can we use this to predict actually the regulatory success in this other task. And this other task is slightly different. So, but it has, I mean, for a lot of people, it actually is quite identical, right? So, but it has some different features. So here, we don't ask much how much you want to eat. We ask about how much you want to pay for eating this item, for your right to eat this item. And then also, we don't tell them focus on health or focus on taste. We tell them, look, we want you to do whatever you need to do to distance yourself from it so that you can regulate or down-regulate your cravings. And what we also see here when we do this out-of-sample predictions in this other data set of 32 participants, using our model from data set 1, we can actually predict quite well with an even higher correlation coefficient the behavior, so the self-regulatory behavior. This was the first study which shows that there's something in the anatomy of our brain that actually helps us to predict individual differences in ability to make healthier choices when faced with a conflict. Now, the next study, now I'm moving a bit back and forth between these different types of self-control in the money and food domain. But this one here is now on the idea of, can I also have a predictive marker? So here we do prediction in the sense of a machine learning algorithm, where we're interested in whether we can actually predict this delay discounting parameter. So people's ability to wait for larger later rewards. And this is joint work with Joe Cable and Carrie Lehman and Bernd Weber and Marie-Christine Simon, and was spearheaded by also my former postdoc, Ioni Koban. So, and so what is interesting now when we switch to this related behavior of individual differences in discounting, we know actually that, which is a bit similar to what I just showed you for the food stuff, right? That, when we look at structural measures, also, so this idea of brain meta density, yes, there are also regions, actually, interestingly, similar brain regions in the ventral medial cortex and temporal parietal junction, but also in the those lateral prefrontal cortex that do predict people's ability to wait for rewards. And then there's other work that also looks at connectivity between brain structures, so looking at white matter connectivity. And same here, they also find that quite similar regions, specifically focus here on the lateral prefrontal cortex, are predicting, so the connections between that and the brain's valuation system does also predict delayed discounting. Here, what is less known is, so these are all similar to what I presented to you earlier, are studies looking at brain structure. What has not been looked at so far is whether there's a predictor based on functional data. Like multiple studies have looked just for correlates of this, right? We are interested to develop in this project a more transdiagnostic marker actually of discounting behavior that also can, but I think I don't have this in my slides, but I can already give you a spoiler alert. So it also predicts more consequences of reward impatience. So it helps us also to predict, for example, BMI and some other metabolic or other health indicators, insulin resistance, for example. So, sorry. Now what we, the question in this study was like, okay, can we actually develop such a multivariate brain marker? And so for those of you that are used to brain imaging, so again, the difference here is that it's not a univariate approach. This one is really a predictive approach using multivariate approaches and machine learning to develop a marker that looks simultaneously not at one or two brain regions or how they're connected, but at the whole brain and how they are correlated actually in some ways. To predict the impatience. And what we did is here, similar to the study that I presented before, is that we had two data sets. One we used to train the model, one we used as a out of sample independent test to predict then, or to test the model, see whether it also predicts in a different study set. The first study was a study that we conducted in Germany, where we had 112 male participants that were doing a delay discounting task, where you saw both of the items on the screen, and you had to choose between either the sooner, smaller, or the larger, later reward. And then the test set was both genders. It was very different, actually. It was a younger population. These were Americans, not Germans. And also the task was actually quite different. So there was always a baseline option that you had to compare, and then you saw the other option on the screen. So you like here, the $24.35 as compared to a fixed amount that you would get now, which was $20. And then you could accept or reject. So you see that the task again is, this one is actually much more different than the differences in the tasks that I showed you earlier. And now, so what we did is, so the analysis approach, and I'll be, not going too much into depths of that. So we said, okay, there are different processes we could look at. There's people see the offer, so the decision, the choice, just the choice onset. We could look also for the process that would encode the larger later amount, right? So this would be when I would then put in the larger later amount as a parametric modulator, or I could also look at the role of the delay by putting this in as a monitor. So we took the three first level images and then put them all together in one mask. And then we looked at, we reduced this through like a lasso PCR. And then we found decision weights in different brain regions, right? That then we used to do a cross-validation, and that we could then use to predict and so to do a comparison again between the predicted and the actual log K using the prediction algorithm here. And what we find is that our prediction, I'll tell you a bit more about the pattern in just a second. First of all, I want to speak to the fitness of or the nature of the prediction. What we see is that the prediction actually works quite well, right? So we see here that there's a correlation coefficient of 0.45 in the set, in the cross-validation set, but then also in where we were very excited to see that in the second data set, so in the out of sample prediction, we were also able, although this was such a different study in such a different population, we were able actually to do a very decent prediction based on our brain model. Now, how does this marker look like? To give you, for those of you that are more interested in the underlying brain function. But what's interesting actually is we have all these hypotheses, obviously, that the brain valuation is involved. We have this hypothesis that memory is and episodic memory, but also working memory is important for in future planning abilities, are important for this idea of delay discounting, and obviously also cognitive control regions. And when we now look at, so these are now thresholded maps for each of these different components, right? We actually do see that the brain valuation system does play an important role, but something that we have not paid that much attention to is this conflict monitoring system, so the dorsal anterior cingulate cortex. But also we see also here the DLPFC again, but we couldn't find much evidence that memory systems are actually involved in this multivariate marker. Which is also interesting for trying to understand what are actually the underlying processes for delay discounting. And so and we tested this a bit more. I mean, this is not this is more like a descriptive test, I would say, right? So we overlaid this. What we find, we searched for different terms for value, reward, and emotion and affect. And what we find is now we can see how much overlay is there for the choice part of the marker and the part that encodes larger later rewards and that encodes delay. And what we see here is that there's specifically for the choice onset there's a positive spatial correlation and there's a negative spatial correlation for the two other parts. And we find that delay, when we look at this one here for conflict and control, that this is specifically, there's a negative spatial correlation for the delay part of the marker, and the other two are positives. And again, where we find less of evidence for an involvement is the idea of memory, imagery, and planning. So, but again, this is obviously a bit more descriptive to interpret this marker. Now, next, again, what I was saying a little bit in the beginning is also that, so why do we do this, right? So because obviously the moment we have the functional data available, we also have their behavior. So why predict the behavior with something as complicated as the marker? We do think that this gets at something that is underlying a lot of other more downstream consequences also of reward impatience. For example, here, when we look at BMI, we do see that specifically actually in study 1, we can predict BMI or correlate this with BMI quite well. Study 2, a bit less so, but also, I mean, obviously this was not set out to predict BMI really, right? So, but then if we look at BMI groups, so as you see in this lower part here, right, we do actually see that for both studies there are significant differences in the K-marker response for people that have a BMI higher than versus lower than 25. So there seems to be something in there that also does predict downstream consequence. Again, we have this also for other measures. So the interim conclusions here is what I showed you for the moment is, well, yes, in the brain there are actually individual differences that can be explained by looking at brain anatomy for food choices, right? Specifically, the gray matter and the VMPFC and the dorsolateral prefrontal cortex matter for regulatory success prediction. Then looking at the related behavior, so looking at self-control for money, in other words, can also be predicted by a distributed pattern looking at functional data. And that this pattern actually, recruits similar structures than we see in the structural work. Not only my structural work on the food choices, but also structural work that actually looked at delay discounting, right? So in the brain valuation system, but also actually regions involved in conflict detection and cognitive control. And also, so we are trying to develop this now. If you are working on fMRI data, I guess not a lot of you are actually working on fMRI data, but if you have done delay discounting tasks, we're very interested in generalizing this marker further. So if you're interested in teaming up and sharing the data, I'm here. Let me know. Now, let me maybe pause for questions. Do you have any questions for this part? Because now we're switching a bit gears. Okay, sorry, I should have checked the chat earlier. LL stands for larger latent rewards. Sorry, Wojciech. And SS stands for smaller sooner reward. Yeah. Is there any other questions? Yeah, Chris, you have a question?
Yes, I do. Thanks very much for that so far, Hilke. It's really, really interesting. So my work looks at the role of context in influencing the foods that we choose and the portion sizes that we choose, and more specifically, the sort of more cognitive side of the context. So the ideas that we create about what breakfast time is and lunch time is. So I was just wondering, and it might just be speculation that you can provide, but if you have anything data-wise, that'd be great. But do you think the self-regulatory behaviors, as you sort of showed, assessed by the grey matter volume or other markers, do you think it would have a similarly as good a relationship if the sort of the healthy or the tasty foods that you offered were presented out of a usual context? So if those foods were presented, let's say, I always use pasta at breakfast time, but that's a very UK specific example. But if it was presented out of that context, do you think the incongruency of the food to mealtime association would fire up another part of the brain and actually muddy that relationship at all?
Yeah, I mean, this is speculation, but I think, I mean, you could actually use such an approach of a signature, maybe, you know, in this case more for food. You could actually use this to predict the differences in context, right? So I think this would be actually quite interesting to do, and I think it's a super exciting question, because also I think so. I mean, at least, you know, I mean, this is now Europeans trying to think about why in the United States people have a bit higher waistlines, right? It's always said, so at least in France, I don't know that much about the UK, we restrict eating times, right? So we say, this is breakfast and you only eat this and you don't eat anything else, right? So, and you can only eat breakfast actually at 2 hours a day. You can literally only get lunch in most of the French restaurants for 2 hours a day. So it's not that you can get it everywhere. Obviously, I mean, there's McDonald's and whatnot, right? But there's still in the cultural, so this is a bit more of a sociological explanation for the French paradox, right, is that there is this notion of certain eating times and cultural norms around them. And I guess that includes also what you eat, right? So I think that's a very cool, interesting question. If you have any results to share,
So
please
these are really important points, I think, but let's save more general questions for later. And I think if there are any clarification questions, that'd be okay, but it looks like you already addressed some of those things. So I would suggest we go on and then save further questions for later.
Yeah, cool. Yeah. Just one thing that going back to, so there's one thing in the chat that I think directly relates to the brain marker. So we would love actually, so this is one of the next steps. We would love to apply this also to eating disorders. We're in the process of applying this to participants with severe obesity, but also trying to do this for people with eating disorders. So you have to stay tuned for that. But you know, this is basic research. We hope to have some actionable insights of this for also eating disorders. Although, I mean, fair enough. So what I'll show you now is more looking at obesity actually. So, and this brings me directly to the next slide, right? So one thing that, so I got to this, you know, alliance that we have with Sorbonne University. I did get my hands on our nutrition science department and the doctors in there, and they're really super excited about doing this type of research. So they allowed me access to their participants with severe obesity before and after they actually have get gastric bypass surgery, and I will show you a couple of results from that research effort. But so the reason then why, so what I learned from this is actually that there's another organ that is very interesting to look at, and that is actually an independent nervous system. So it's our GI tract, so our gut, in other words, and so this is interesting because the gut actually, so first of all, there are obviously lots of chemical signals that are involved in signaling the brain, for example, hunger and satiety. So it is related to our reward system, and specifically the neurotransmitters dopamine and serotonin. But also there's something else that, and that I got really excited about to look at is a bacteria in our gut. So this is actually not us, this is somebody else living in us, right? So the bacteria, and they, and it's, I mean, in some ways it sounds a bit scary, right? Because they have this power to actually, either through these neurotransmitters or other chemical messengers, to influence our brain, right? Our signals in the brain, and even through neuronal pathways, so the vagal nerve, right? So that would be involved in sending neuronal signals. And what is interesting is that the gut microbiome is influenced by what we eat, right? So because a lot of research looks at, you know, can I look at the brain and predict what we eat? But this now reverses this a little bit. Can actually, the consequence of this, so what I eat, have a feedback loop because of it might change actually the gut microbiome, that in turn will change the processing in my reward systems and other related systems, and through that also have an impact on my decision-making abilities, right? So, and we know so this is obviously very new work. A lot of work is correlational, and we do have evidence that it's linked to certain health outcomes. So a rich microbiome, for example, is usually related to good health, right? So for example, participants with obesity, or patients with obesity, they have a low diversity and a certain type of composition. It's also involved in stress and mood responses and mental health, right? And this was then, this is a more complex version of what I was trying to say earlier. I said, look, there's actually lots of ways on how the gut microbiome can influence our decision-making abilities in the brain. What is interesting now is that a lot of these studies, as I said, they're correlational, and a lot of the more causal studies, they're really done in mice, because you can have them with and without bacteria. So you have germ-free and not germ-free mice. And so I wanted here to look at, to get a kind of a quasi-causal relationship by looking at an external shock, so an endogenous shock to the gut microbiome system, so an intervention. And this is either, so the next two studies I'll show you are on gastric bypass surgery, because what we know is that this gastric bypass surgery does improve the gut microbiome diversity and profoundly changes the gut microbiome composition. So I use this diff and diff approach, looking at them before and after versus control populations that didn't undergo the surgery. And then in the last study, I actually look at the other way on how to influence this, would be to look at a dietary intervention where people are eating something that grows or changes the composition. What we did here is looking at the administration of symbiotics, so probiotics and prebiotics mix. And so the first two studies, again, that are looking at these participants before and after gastric bypass surgery and that have severe obesity, is joint work with the nutrition clinic at the Sorbonne University, so with Karin Clément's group. And here, so what we did here is to make the link between not necessarily the biome directly, so this indirect link that we're making through this intervention that we know that changes this, but it also changes some of the other parts of the graph that I just showed you. For example, these hormones that are signaling hunger and satiety, specifically leptin was what we focused on in this study. And what we when we think about the role of the brain valuation system in obesity, we know, so I mean, I showed you already that neuroanatomical markers are important for dietary self-control. But here, when we look at really participants that have obesity, what we know is that specifically now I'm focusing on activity at rest, right? So before I showed you task-based activity, here I'm showing you what actually happens when you think of nothing. So we call this resting state connectivity. What we do see is that, well, if you look at compare participants with obesity and those that are lean, you do find in the brain valuation system differences in the resting state connectivity. And also connectivity between the thalamus, the caudate, and the VMPFC are predictors of BMI. And this is work that looks at resting state, but also looks at structural connectivity in the white matter, what I already talked about briefly earlier. But what we try to do here is really to look at, can we predict change in weight over time, looking at these regions that, you know, now I'm very interested in, so the brain valuation system and the cognitive control system. And what we're doing is, so we're setting the seat in the VMPFC, and we look at connectivity to those other two areas and see how they change. And now, so what we did here, so the study, we had 44 lean participants and 14 participants or patients, to be a bit more precise, with obesity. And what we did is, so we had them at baseline and we had them 8 months after gastric bypass surgery. And what we also did is we sampled a bunch of systemic measures, and specifically we looked at leptin. So this is a hormone that is involved in signaling satiety. And what is interesting is that, so similar to insulin, actually, it's for patients with obesity, it's actually dysfunctional. So it doesn't really work properly. It's locked at a very high level, but it doesn't fluctuate anymore. So they have something that, you know, medical doctors like to call leptin resistancy. Whereas, you know, healthy participants, for them, that obviously that works, right? And then we have this measure of connectivity at rest within the brain valuation system and also the cognitive control system. And so what we did here is, so we looked at, first of all, regions, taking the hotspot of the valuation system as a seat, so the VMPFC as a seat, and look at whether there is a difference between the lean and obese, so whether there's sensitivity to obesity. And what we find, and this is at the first glance, it might actually be unintuitive, because so we see actually that the obese versus the lean, they actually have a higher. Sorry, hold on, there's something bugging on my computer. Yeah, so they have, what you see here is, so these are the obese before surgery. This is actually the connectivity, between the VMPFC and the DLPFC is actually higher for the obese. So this would speak for that they actually, so their evaluation system is better connected at rest to their cognitive control system. And for the leans, this is worse. And this difference is actually significant. However, the interaction is not significant. So the obese at both time points have more connectivity there, right? So, and then, so the second region that we found to be sensitive to obesity is within the brain's valuation system. So this is the ventral striatum now. And here we find also something that you might think at the first glance is unintuitive, because what we find is that here the obese have actually lower connectivity within the reward system. And this improves to the level of the non-obese participants after the surgery. And the lean controls say, I mean, it goes a bit down, but it's not significantly different. And so this, again, and this is why is this unintuitive, because specifically from a layman's perspective, you think that, you know, participants with obesity, they have a dysfunctioning reward system. So you could have predicted the opposite. But there's this dopamine deficiency hypothesis actually saying, like, look, it's actually that in some ways, to put it simply, meaning that the dopamine neurons are blunted, right? And that's why for the same amount of pleasure, you need much more stimulation. And this is in some ways in line with this idea, what I'm showing you here. So we do find that these two regions are actually sensitive to obesity. And what we do now is we extract the connectivity at both time points in these regions, and use this to predict actually the change of weight over time. And what we find is that here now this is VMPFC to ventral striatum, so within the reward system, connectivity at rest, we do find that this actually you know, predicts the observed weight loss quite well over time. And then when we do this, okay, no, so sorry, this is not what I want to show. So when we do this for the DLPFC, actually it doesn't work this way. So there's no predictive power in using that resting state connectivity. So it's only true for the VMPFC to ventral striatum resting state connectivity. And what is interesting is now, and this is only for the patients with obesity for which we had leptin data before and after surgery. This is correlated actually with a change in leptin. So their improvement in leptin resistancy seems to be related to this change in resting state connectivity in the brain's valuation system. And this is now interesting because we know, I mean, again, there's not a lot of causal evidence and we're far from providing any causal evidence here. But the idea is what leptin also does, other than signaling satiety, it's also indirectly linked to the dopaminergic pathways. And the same is true for the brain's valuation system. So there seems to be, in other words, again, this is so Wolfram would now yell at me because obviously I'm not measuring dopamine here, but we all like to talk about dopamine. So the dopamine seems to play a role here. So it would be actually cool to work with someone like Wolfram where you can actually measure activity from dopaminergic neurons. But so we do think that this plays a role here. Right? So although we only have indirect evidence for that. Now, so in the next study with the same co-authors, we now looked at what actually happens when we look at this idea of reward impatience. And here we did money and food. So a bit more similar to what I did with my kids in the beginning. Is that also affected by this surgery or not? And we did this in 2 populations. In the first population, this was a very simplistic behavioral task where we asked them, do you prefer receiving 4 candies now or 4 in a week? And then we varied the distance and time, so the delay. And what we find is that now these are a similar patient population. So we have patients with obesity before they do the surgery, the lean controls, also twice in time. Here the timing was just a bit different, was 12 months apart. And what we see is that before surgery, they do, the participants with obesity, they're much more impatient for food rewards. And this kind of gets them closer to the level of the lean after the surgery. And then we tried to replicate this in a study where we have a slightly different delayed discounting task. What we see there, so we vary the amount of foods that they can get later and also the delay when this later is. And very similar here, although it's not, the interaction is actually not significant at 0.05, only at 0.1. What we find is that same as before, so there's a difference at baseline, and this difference tends to attenuate after the surgery. And when we do the same for money, we actually don't find evidence for this. So this seems to be specific for food. And so we've done actually a lot of other tasks in the, so other kind of behavioral tasks in this participant population with obesity. And what is fascinating is that this surgery really, so it really changes the metabolic profile linked to the state of obesity, really changes a lot of decision-making stuff. Also how much I am, I'm prone to, for example, respond to marketing activities of food companies. So if I fall for certain biases, based on marketing actions of the food companies, right? And the surgery seems to do actually a lot more than changing the size of the stomach. But this is, we're trying now to explore this a bit more systematically to get at all the other measures that are in between. So this, unfortunately, we only had the behavior and nothing else. So this is why we then ended up setting up the study here. That is still a study that we're in the midst of analyzing, where we looked at, so it seems from this other data that the biome seems to, the biome and also other systemic markers in the blood seem to matter for how my brain actually makes those good or bad decisions or self-controlled decisions. And so again, what I showed you mostly is that, we know a lot about how the brain encodes this, right? But how what we eat actually changes, and in turn, decision-making we know much less about. There's some work actually specifically from Molly Crookett's lab, looking at when I, for example, look at serotonin, so which is a neurotransmitter that works in some ways the opposite from dopamine. If I deplete this, sorry, my thing here is just moving forward. This actually changes behavior. For example, social behavior is something that you know much more about than I do. So for example, fairness behavior in an ultimatum game. So it changes, increases, the tryptophan depletion increases the rejection rate for unfair offers. If I put you on a diet, breakfast diet that is high in carbs and protein ratio, that also increase rejection rates to unfair offers and changes tyrosine levels. And then so some of this also has looked at impulsivities or delayed discounting. So tryptophan depletion increased impatience for money, also Molly's work. So we were interested here, does it also extend to intertemporal choice, and specifically not looking at tryptophan depletion, but looking at dietary intervention that would change the gut microbiome composition temporarily, right? So this is clear, not something that will do this in the long term. We said, people, look, we don't want you to change your dietary patterns profoundly. We want you to just take this supplemental pill to eat whatever you eat normally. But they did get a dietary supplement that was a symbioticum, so something that the bacteria they feed on, but also that contains them, right? So because otherwise they don't colonize really in the gut. So our prediction was that this intake would actually have an impact on the dietary composition and also on the composition of the gut microbiome. And then we pre-registered that we would think that because this is linked to improved health, that it would also improve decision-making, in this case, make them more patient. And then that this works through differences, that it alters the underlying brain processes. And this is what I, just explained to you. So we had 100 male participants. They came to the hospital at Bonn University. They were actually very, so they had to give stool probes to get the microbiome data. But they did a lot of other behaviors, and we also draw blood from them. I will not talk about that part, but it's very complete, right? So this is what we're looking at right now. And then they did an Fmri task. And so then after that, half of them were randomly assigned to a treatment group that did receive the symbiotic supplement, and the other half were assigned to a control group that did get other supplements that looked identical, but it didn't have the pre or probiotics. And then we scanned them again, and we're interested again in this before and after. To give you some more background on this stuff that we gave them, so it's a mix of probiotics that are actually good bacteria and prebiotics that helps them to colonize, helps them to survive in your gut. And so what was nice is actually that we convinced the manufacturer of those commercially available products to also produce a placebo for us that looked identical, had the same packaging and so forth. And then, so then we used the task, the same task I explained before, where we asked them to choose a smaller sooner versus a larger later option, and we varied them systematically, as you see in the table here. And again, so we're interested in this K parameter, and the higher the K parameter, which is the discounting parameter, the more impatient they are. So the higher the K, the more impatient you are for rewards. And first of all, so the first question, getting back to our hypothesis, was, does this intervention work? Does it actually change the abundance of the bacteria that we gave? And luckily the answer is yes to this. So we do see that it impacts the abundance in the symbiotic group, but not in the placebo group. Now, what is interesting is, and this is now where, if you have any thoughts on this that go, that will help me to explain this. We do find a significant difference also in the impatience, but we find it in the other direction as we predicted. So we predicted that this would help them to be more patient, but we find that it makes them more impatient. Right? So this is in the symbiotic group, but not in the placebo group, and this interaction is significant. A different way to think of this is that you can plot these utility curves, right? So, and what the K actually is expressed in the slope of these curves, right? You see that for the placebo groups, these curves are identical, whereas here you see that there's a difference actually in the slope of the curve, and this is significant. So we spent a lot of time figuring out what's going on there. So again, if you have ideas, very welcome and excited to hear them. We looked at, so is there any differences that happened, although we randomized, right? So in sociodemographic differences, there's no. Did they actually maybe know that they were in the treatment versus the placebo group? No, that also did not affect our results. Are there any other differences at baseline on BMI, mood, risk preferences that are related to delay discounting, ability to reflect, so use a more cognitive systems or the cognitive reflection task, perceived quality of life or rationality of choices, and all of these, there were no differences actually. So that unfortunately did not turn into an interesting explanation. The only thing that we're looking at now is, and this was a bit of a surprise to us to see, is now this, for those of you that are not microbiologists, which are most of you, this is a bit Chinese here, right? So let me explain this to you. So these are kind of the 4 different bacteria that we gave them. And what we do see, and this is what I showed you earlier, just in a different visualization, is that, so these, the abundance of those becomes positively higher, specifically actually this one here, after the intervention. But also what happens is that there's obviously there's, there's interactions with other bacteria. And what we see is that, it increases also all these other bacteria, and it decreases, all of those, right? So there seems to be something much more complex going on that we did not predict it to be that complex. And these different bacteria are differently linked also to dopamine and serotonin. And we're trying to, understand these correlations a bit better now, to understand why we actually have an effect in the opposite direction as we would have expected. Now, the last thing I would like to show you is that, so we also then looked at, so we have also Fmri data from these participants, and we used our marker that I told you about before. And we looked at, does actually the intervention affect the brain marker? So we can look at this before and after and compare this with the placebo group. And what we see is in the intervention group, it actually, yes. So this is now the treatment group that received the symbioticum. We do see an impact on the delay discounting part of the marker, on the delay encoding part of the marker, right? So it seems to alter the underlying processes for encoding the delay of the choices that they're doing. And we don't see anything like this in the placebo group. And then I think maybe let me check the time. I think maybe for the sake of time, we skip this. And we also find some stuff on when we look at markers of serotonin and dopamine, but again, this is exploratory for the moment. So yeah, and these are the next step, is to understand this a bit better, and then also, to put this into relationship with all these other markers that we're having. And also for the behavior, we have not looked at some of the other behaviors that we sampled, for example, sensitivity to gains and losses that has been linked to delay discounting and so forth. So, and again, if you have any thoughts, let me know. So to conclude, what I've showed you today is that individual differences in our biology matter to explain self-controlled behavior for money and for foods. Specifically, I showed you that in the anatomy, so the gray matter density in our reward or valuation and control system can be used to predict weight loss, but also can be used to predict ability to make healthy choices. Very similar regions are involved in a marker predictive of impatience for money. I also showed you that resting state connectivity in the system is important to predict weight changes over time, and specifically actually resting state connectivity within the valuation system, so between the VMPFC and the ventral striatum, and not the DRPFC as you could have thought, right? Does predict weight change over time. And then I showed you that participants with obesity are more impatient for food as compared to lean, and this is not the case for money, and that something that really changes the activity of the GI tract, namely the gastric bypass surgery, decreases this in patients, so makes them more similar to the lean participants. But a symbiotic treatment that changes in a more subtle way also the gut microbiota diversity and composition leads to the opposite result. It increases impatience for rewards. And with that, I thank you very much for your attention, and we have time for all of your questions now.
Thank you so much, Hilke. This was absolutely fascinating. I must say, how you bring together all these different aspects of eating behavior. I should say to our audience, we have a bit less time than usual. We have until 5:45 for questions. So we probably should jump right in. So if you have any questions, let me know. Camilla Nort.
Hi, thank you so much. Such an exciting talk. I really enjoyed it. And my favorite study was the gastric bypass one. I thought it was a phenomenal idea to study that particular population. What I'm curious about is why you hypothesized that, at least in my understanding of the talk, to be the gut microbiome driving any effect you found, because my understanding of gastric bypass surgery is that it could just as well be an effect, for example, of brain synchrony with signals from the stomach, which are sort of well known, and it could be that disruption caused the neural effects that you found.
Yeah, there are lots of things that could explain it, right? So that is true. But I mean, we know that what the gastric bypass surgery does is, for example, changing insulin resistance, changing leptin resistance, but it also changes the diversity of the gut microbiome and its composition, right? So this is a bit, so I got excited about this idea, and this is why I set up this other study like this. There's also very interesting other effects. So for example, what you just said, the synchrony, and specifically, for example, also the vagal nerve communication, right? So, and this is something else that I would like to explore, but that's, I think, a question for another research study. But I do agree that there could be all sorts of other explanations, right? So, so this is one that I wanted to better understand whether this is, this idea of the gut-brain axis playing a role here would actually be the explanation, because obviously it's a topic specifically my colleagues from nutrition science are very excited about. So, and it would be wonderful if that could help us to better understand disadvantageous behavior.
No, of course. Thank you.
Yeah.
Next, we have Olivia Stupar.
Hi, thank you. That was an amazing talk. Really, really interesting. I was wondering, did you collect the data, any health data or nutritional data in the final study with your supplement versus placebo group, in terms of their potential BMI at the end of the study? Because in patients, with things like Crohn's disease or ulcerative colitis, quite often a medical treatment is to increase the diversity of their microbiome in their gut for optimum nutrition. So I'm just wondering if maybe when you've disrupted this microbiome so that you've got this predominance of these four that you've given, are you getting some difference in digestion so that they're getting different nutritional standards, and that's changing their set point, like you mentioned with the obese patients having a set point difference earlier?
That's an interesting question. I don't think that we measured this. I mean, we measured a lot of the blood markers of, for example, liver and fat and fat enzymes and so forth, right? So, but we, for the moment, I mean, so first of all, we don't find any difference so there are no differences in weight status or weight or body fat. That's what we looked at, right? So, but we also didn't expect it because it's such a subtle thing, and they only did it for 8 weeks, right? So it would be interesting to see, for a longer period, whether this would change weight, right? So, but I think so. How would you measure what you were asking about? Maybe my lack of understanding of these processes, would help me to answer your question. So, how would I measure this? What you were referring to?
I'm not sure, actually. It's not really my area of research. I'm a medical student doing a PhD in psychology, so there's relations there. And I do think you'd have to do something quite a lot more invasive in terms of
no, that we didn't do. And it was very hard to get participants. Right? So no, I mean, we know we had there's some trends, and again that it changes how the liver is processing, for example, fats, right? So, but that's still, we're still looking into that. We also found some evidence that it changes a bit. So we asked people to tell us these nutritional diaries, right? So they tell us
Mhm.
for 3 days a week what they're eating, but this obviously, I mean, this is hugely flawed because you know, you have to weigh things, and right, so there's lots of reporting biases, I'm sure. But then we didn't find that it does affect the eating habits, right? So yeah, but these are definitely very interesting questions.
That's
But again, I
interesting.
mean, I think you have to keep in mind that this was a very subtle intervention, right? So
Yeah.
yeah.
Okay, thank you.
Yeah. And there are also a couple of things in the chat. Simona, should
Yes,
I?
yes. We earlier had a question from Ikim. I don't know if Ikim still wants to ask that. That was kind of halfway throughout the talk. So that was about, let's see, how the task-based fMRI.
Yes, I think this was related to the resting state study, right? Yes, it hasn't been really done to use this to predict weight changes, but there's some work that looks for correlations, and they also find that activity and reward structures would be correlated with this, right? And there's a lot of work. So nutrition scientists, when they do fMRI, they like, rather than having a decision-making task, they like this cue reactivity task. So where you just expose people to food in the scanner and you look at what's happening in the brain. And they also always find that there's regions involved in reward that differ between obese and lean. But I mean, so what I find interesting about these studies looking at people, there's the same participants before and after the gastric bypass surgery, is that there's this layman intuition that, you know, you're obese because you have problems in self-control. But the study that I showed you with this, with the discounting of food shows is like, well, maybe so there is, we can find that there's a difference between the obese and the lean, but if it's not something that is a personality variable, right? So it's something that could be linked to your metabolic profile, and this is why I'm interested in these individual markers on the biological level, and not so that's linked to the state of being obese, right? For example, the leptin that I showed for the obese participant in the resting state, right? That actually is that is causing this. That I maybe make choices where I prefer more food versus less food, right? So, but it's not something. It's not a personality trait of mine that I'm bad at this, right? So it's a much more complex story. So it seems to be something that it's in my metabolic state, much more in my willpower, right? And I think this is important for the obesity community.
Yeah, I agree. I think this is really, really fascinating that it may not be about willpower at all, right? I mean, I also think that study with the surgery was just so fascinating, right? Because from what I understand, all that happens is that the stomach becomes smaller, right? So why should that change the decision making? So that's related to food. So that's quite remarkable, actually. So yeah, very interesting. I think we have, let's see, we have a few more minutes if anyone else wanted to jump in at that point. If not, then maybe I'll just share a very quick thought about why that might be that well, I couldn't really say I have an explanation, but here is just a thought in general.
Yeah.
So is there any way that having, you know, a diverse gut biome actually means one has to get a certain range of nutrients, or some, you know, you need more of something. Basically, you become hungry for certain things, just the range of nutrition and so on that you need rather than the quantity. So is there something that could be there?
Interesting. Yeah, that's a good question, actually. I don't know, but I will ask this to my nutrition scientist colleagues in my research team. Yeah, I mean, it's also, I mean, for me as a, you know, as a person trained in consumer psychology, it's fascinating that for now all the research points to the more diversity the better, right? It needs to be some inverted U shape, right? So there needs to be a point where maybe this is not the case. Although I mean, it's clear that with a subtle intervention we're not hitting this point, right? So, yeah, it seems that it also does something else, right? And I think your question is an interesting one. The problem is also that it's not there's lots of work in animals, there's not a lot of work in humans, so we might not even know, right? So, but maybe, I mean, this is, I think what you're suggesting could be seen in an animal population. So that's an interesting thought. Yeah, thank you, Simone.
Right, well, good
Cool.
stuff, good stuff. So we're just about on time. I know that you have to leave earlier. So thanks once again for such a fascinating talk. It was a real pleasure, and I'd love to catch up sometime and chat about this and find out more details about what you've been working on. And in the meantime, thanks to everyone else for joining us. Do join us again in the future. We have these Zangwill talks on Friday afternoon, so keep an eye out on the mailing list. So yeah, thanks everyone. I'm now going to close the call.